Epidermis is present both on the upper and the lower side of the leaf. So if you remember, in dicot I told you the size of the vascular bundle depends on the size of the veins and the midrib, right? They are made up of two types of parenchyma cells, palisade parenchyma cells and spongy parenchyma cells. In the monocots, the stomata are present both in the upper and the lower epidermis. No changes there. Hello everyone, a warm welcome to the session on class 11th biology for CBSE. I'm Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. So in this session, let us study about the anatomy of dicot and monocot leaf, wherein we'll learn the cells that are present inside the dicot and the monocot leaf, and also we'll learn some of the MCQs likely to appear from this particular topic. So starting with the dicotyledonous leaf. Here dicotyledonous, it is also called as dorsiventral leaf. So why it is called as dorsiventral? So it is called dorsiventral because the arrangement of cells that are there on the dorsal side and the ventral side is quite different in the case of dicot leaf. So in the exam, if they ask, write a note on the TS of dorsiventral leaf, don't get confused. It is nothing but dicotyledonous leaf itself. So what are the different types of cells that are present? So it is first covered by a layer called as epidermis. Epidermis, again epi means outside, derma means skin. So the outermost layer, which is the epidermis. So epidermis is present both on the upper and the lower side of the leaf, because for a leaf we can say it has an upper side and a lower side, right? So the upper side is called as the adaxial side and the lower side is called as the abaxial. Now how can you remember this? It's quite confusing, right? Adaxial, abaxial. So lower means always remember it as back. So therefore lower side will be abaxial side, upper side will be adaxial side. So therefore covers both the upper surface, that is the adaxial epidermis, and the lower surface which forms the abaxial epidermis. And the leaf has a conspicuous cuticle. The cuticle, especially in the case of the leaves of hydrophytes, that is those plants that live in water or those plants that live in deserts, as a term of modification, they have thick waxy cuticle in order to, say for example in hydrophytes, in order to protect the internal cells from damage that is caused by the water, because hydrophytic plants always they are in water itself. So this thick waxy cuticle acts as a water repellent. And in the case of xerophytes, that is those plants growing in the desert, those plants need to save water. So water should not get transpired from the cells. So to prevent that, around the epidermis, they'll have a thick waxy coating. So that is about the cuticle. So the abaxial epidermis generally has stomata. So stomata are usually present in the leaf. We have studied about the stomata while studying about the epidermal tissue system in the first session of this chapter. So you can go back and have a look at it if you get confused. So what is stomata? Stomata are nothing but bean-shaped structures that are present on the leaf, which helps in the process of transpiration, exchange of gases and all that. So the abaxial epidermis, that is the lower side of the epidermis, has more number of stomata when compared to the upper side or the adaxial side of the epidermis. Sometimes in the adaxial side, in most of the plants, they do not have stomata at all. So therefore, stomata is concentrated in the abaxial side or lower side of the epidermis. The adaxial epidermis, sometimes as I told you, they may even lack stomata. They will not have stomata at all in most of the plants. So we can see here the stomata that is present. It is usually distributed in the lower side, and they have the adaxial epidermis which forms the upper epidermis, and they have the abaxial epidermis which forms the lower epidermis, and it is between the upper and the lower epidermis that the cells are being distributed. And this entire thing from here till here we call it as the mesophyll. It's called the mesophyll of the leaves. So what is this mesophyll? Now, if you look at the arrangement of the cells or towards the adaxial epidermis and the arrangement of the cell towards the abaxial epidermis, you can see that the type of cells that are there are different, right? So that is why the dicot leaves is also called as dorsiventral leaf. So here talking about the next structures. So we'll talk about the mesophyll, that is below the epidermis there is the mesophyll. So far while talking about root or while talking about stem, you might have seen we using the word cortex, right? But in the case of leaves, we don't use the word cortex. Instead, we use the word mesophyll. So mesophyll are the tissues that are present between the upper and the lower epidermis, as I told you. So between the upper and the lower epidermis, whatever tissue comes, we call it as the mesophyll. Now, what are the cells that are present in the mesophyll? Let's look into. So mesophyll, they possess chloroplast, and chloroplasts are very, very important in the plants in order to carry out photosynthesis because they contain a pigment called as chlorophyll. And these chloroplasts are nothing but made up of parenchyma cells itself. So usually they are made up of two types of parenchyma cells, palisade parenchyma cells and spongy parenchyma cell. What is the difference? So look at the palisade parenchyma cells. They are elongated and they are arranged close to each other without much intercellular spaces. But look at the spongy parenchyma. Can you see these are the spongy parenchymas that are there? Spongy parenchymas, they are of irregular shape. They do not have a particular shape and they are arranged with large intercellular spaces. And palisade parenchyma are located towards the adaxial epidermis and spongy parenchyma are located towards the abaxial epidermis. And there are a lot of intercellular spaces created between the spongy parenchyma, which is also called as the air cavity. So this is about the spongy parenchyma. So therefore, they are made up of two types of parenchyma, and both of these parenchyma, they contain chloroplast in them, which contains the chlorophyll pigment, which brings about the photosynthesis. So this is about the mesophyll cells. Next, moving on to the vascular system. Again, here vascular system, it consists of the vascular bundle, which is made up of the xylem and the phloem. Now, where are the vascular bundles distributed? So you can see the vascular bundles in the case of leaves, especially in the veins and the midrib. So when you take a leaf, draw a leaf, this will be the midrib. This is the midrib and all the lateral veins arising from it, this is the veins. So midrib is the main vein from which all the other veins arise. So vascular bundles can be seen wherever the veins are present and wherever the midrib is there in the case of leaves. And the size of the vascular bundle depend on the size of the veins. So some veins are very, very thin thread-like. They'll have smaller vascular bundles. Those veins which are thicker and bigger in size, they'll have larger vascular bundles. So always it depends on the size of the vascular bundle, depends on the size of the veins. And the veins usually vary in thickness in the case of reticulate venation. So what is reticulate venation? So it will have a midrib veins, and these veins will give rise to a lot of branching like this. And as and when the number of branches increases, the thickness of the vein also decreases. So therefore, those leaves having a reticulate venation will have different sizes of vascular bundle distributed in them. And the vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. So, if you can see the vascular bundle, they have the bundle sheath cell which is thick-walled. So, this is this layer. It is called as the bundle sheath cell. We can't call it as the endodermis here because it surrounds the vascular bundle. Their endodermis was surrounding the pericycle, right? It was present just below the cortex. Here, entirely there is vascular bundle. So, entire it is phloem is there inside and then xylem is there, and covering the phloem entirely we can see the bundle sheath. It is called as the bundle sheath. So, moving further, we'll talk about, so that was about the dicot leaf. So we'll talk about the monocot leaf. Monocot leaf is called as the isobilateral leaf because in monocot leaf, if you can see, there is no changes in the cell when we look at the adaxial epidermis or the abaxial epidermis. In between, no changes in the cell. It looks entirely the same. So therefore, it is because here how the venation is completely parallel. So therefore, we call it as the isobilateral leaf. Here also they have the epidermis in which stomata are present on both the surfaces. In dicotyledonous leaf, stomata were present more on the lower epidermis, that is in the abaxial epidermis. Stomata was almost absent in the upper epidermis or the adaxial epidermis. But in the monocots, the stomata are present both in the upper and the lower epidermis. No changes there. That is also one of the reasons why we call it as an isobilateral leaf. And talking about the mesophyll, which consists of parenchyma cells right between the upper epidermis and the lower epidermis. So this is the mesophyll region. Now what is the mesophyll region made up of? So the mesophyll region is not distributed into palisade or spongy parenchyma like how it was in the dicot, right? In dicotyledonous leaf, towards the adaxial epidermis, they had palisade parenchyma, and towards the abaxial epidermis, they had the spongy parenchyma. But in this case, they just have parenchyma cells with intercellular spaces having chloroplast in them, which helps in photosynthesis. There is nothing like palisade or spongy parenchyma in the case of monocotyledonous leaf. Next, there is some specialized cells that are present in the monocotyledonous leaf, which you don't get to find in dicot leaf. They are called as the bulliform cells. So usually grasses is one of the best examples for monocots. So in grasses, when you look at the adaxial epidermis, that is the dorsal epidermis or the upper epidermis, the cells along the veins modify themselves into large empty colorless cells. So these large empty colorless cells that are nothing but modifications of the veins which are present in the upper epidermis or adaxial epidermis of the monocotyledonous leaf, we call it as the bulliform cells. So let's talk about the bulliform cells in detail. What are they? So bulliform cells, they have a particular function. So what is the function? Usually bulliform cells, they help in maintaining the shape of the leaf also. So how we can see? So here, or they help in the process of maintaining the transpiration process also. So when the bulliform cells in the leaves have absorbed water, they become turgid. That is, turgid means they swell up and therefore the leaf surface gets exposed. So here, where are they present? So if this is the leaf, upper side of the leaf will be, so this is a leaf. Imagine this is a leaf in a plant or a twig like this. So entire upper epidermis is there, lower epidermis is there, whereas the bulliform cells present in the upper epidermis. So when the bulliform cells absorb water, the leaves will widen up like this. Why it will widen up? Because the bulliform cells are swelling, they are becoming turgid. So they will store water. Now what happens when the leaves, that is during water stress, whenever the water is lost from the plant, to prevent excessive loss of water, the bulliform cells will become flattened or it will become flaccid, and then it will result in coiling or folding of the leaf surface. Therefore, when folding of the leaf surface occurs, what happens? The surface area of the leaf will be minimized. Therefore, the evaporation of water will also be minimized, or the loss of water will also be minimized. So that is one of the main characteristic feature of bulliform cells which are present in the grasses. You might have seen in grasses and all, sometimes the edges of the grasses will just roll. So that is the reason. That is because of the bulliform cells. So if the bulliform cells loses water, it will become flaccid and it will start to roll to minimize the surface area of the leaf in order to prevent further loss of water during water stress, that is during drought, whenever the water availability is not there and all that. So that is about the function of the bulliform cells. So in monocots, we know that they have parallel venation, right? So I'll draw the leaf of a grass wherein they have entirely parallel venation. So parallel venation in monocot is reflected in the near similar sizes of vascular bundle. So if you remember, in dicot I told you the size of the vascular bundle depends on the size of the veins and the midrib, right? That is during reticulate venation, what happens as and when the branching of the veins occurs, the size of the veins tends to decrease. So therefore in dicot leaves, the vascular bundles are of different sizes, right? But in the case of monocot leaf, they have parallel veins which are all the veins are of the same size. Therefore, the vascular bundles are also of the same size. So this is about the dicot leaf. So now we know the difference between the dicot and the monocot leaf. So let's move on to the MCQs. In dorsiventral leaf, meaning what does dorsiventral leaf mean? It is nothing but dicot leaf. Stomata are abundant in dash side of the epidermis. So we know that it has the stomata which is not equally distributed. Upper side will not have the stomata, lower side will have the stomata. So we'll look into lower, adaxial, abaxial, dorsal. So here we can choose the answer as abaxial epidermis, that is the lower side of the epidermis will have the more number of stomata. Next question, the tissue between the upper and lower epidermis in dicot leaf is called. So there is a tissue between the upper, that is, or they can ask the question as the tissue between the adaxial and the abaxial epidermis in dicot leaf is called. Is it called the hypodermis? No, it's in the case of stem. Epidermis? No, it is the outermost layer. Endodermis? No, it is the layer present inner to the cortex, which we don't find in, that is leaves, because in leaves we call it as the bundle sheath. So therefore, so here the right answer is mesophyll. So whatever cells are present between the upper and the lower epidermis, we call it as the mesophyll tissue. So option D is the right answer here. Next question, which of these is incorrect about mesophyll of dicot leaf? So we have to find the wrong statement about the mesophyll. So here possess chloroplast, yes, that is the palisade and the spongy parenchyma that is present in the mesophyll of dicot leaf. They have chloroplast pigments in them. So therefore this option is not correct. Consists of palisade parenchyma, yes, towards the upper epidermis or towards the adaxial epidermis, they have palisade parenchyma. Next, consists of sclerenchyma, consists of spongy parenchyma. Here, the leaves, neither the monocot nor the dicot leaf, they have sclerenchyma. So, therefore, option C is the right answer here, because this is not true about the dicot leaf. Next question, the elongated cells placed adaxially in dicot leaf. So, adaxially means upper. Or the question can be, the elongated cells placed towards the upper epidermis in the dicot leaf are spongy parenchyma. No, spongy parenchyma are placed towards the abaxial epidermis or towards the lower epidermis. Sclerenchyma, they do not have sclerenchyma at all. Palisade parenchyma, yes, it is palisade parenchyma. So option C is the right answer. Next one, in dicot leaf, air cavities are present in. So we had learned, right, that is the spongy parenchyma that are there. They are distributed with lot of intercellular spaces, which results in the formation of air cavity. So therefore, it is present in spongy parenchyma. The option D is the right answer here. Spongy parenchyma that is situated towards the lower epidermis. So next question, which of these is not true about vascular system in dorsiventral leaf? Vascular bundle size varies. So what is the question? Which of these is not true about? We have to find the false statement regarding the vascular system in dorsiventral leaf, meaning in dicot leaf. So vascular bundle size varies. Yes, the vascular bundle size varies because the size of the veins in the dicot leaf tends to vary. So this option is wrong. Vascular bundles are surrounded by bundle sheath. Yes, the vascular bundles are surrounded by bundle sheath in the case of dicot leaf. So this statement cannot be considered. Vascular bundles present in veins and midribs. Yes, vascular bundles are present in the veins and the midribs. They are distributed in the veins and the midrib. This option cannot be selected. Vascular bundles are similar in size. No, here depending on the size of the venation, the vascular bundle size also varies. So therefore, the vascular bundles are not similar in size. Vascular bundles are similar in size in the case of monocot leaf. So therefore, this is not true about the dicot leaf or the dorsiventral leaf. Next question. The distribution of stomata in isobilateral leaf is, so isobilateral leaf means monocot. So on adaxial, on abaxial, both A and B on dorsal side. It is both A and B because stomata are distributed equally both on the adaxial and the abaxial surface, or the upper and the lower surface of the epidermis of monocot leaf. Next question. Which is not true about bulliform cells? So which statement is false regarding bulliform cell? That we have to find out here. Modified abaxial epidermal cells, modified adaxial epidermal cells, they are modified adaxial epidermal cells. So the statement cannot be considered. They help in maintaining the water content. Yes, by becoming turgid and flaccid and all that. Empty and colorless. Yes, they are colorless cells. So this statement also cannot be considered. Modified abaxial epidermal cells. They are not present in the abaxial epidermal cells, but they are present in the adaxial epidermal cells, wherein they formed because of some of the modifications in the adaxial epidermal cells. So therefore, this option is correct because this statement is not true about bulliform cells. Next question. Which of these is a characteristic of monocot leaf? So here we have to find among the given options which is a characteristic of monocot leaf. Mesophyll has spongy and palisade parenchyma. No, mesophyll has parenchyma, but there is nothing like spongy and parenchyma in the case of monocot leaf. Stomata present in adaxial epidermis. No, stomata in monocot leaf are present both in the adaxial as well as abaxial epidermis. Vascular bundles are similar in size. Bulliform cells helps in water loss. Now, bulliform cells do not help in water loss, but when there is water loss occurring, to prevent further water loss, they become flaccid and resulting curling of the leaf. So, therefore, the answer that is correct here, which is the characteristic that is correct here, is vascular bundles are similar in size, because in monocots, the entire leaf has parallel veination wherein all the veins are of equal size, therefore resulting in equal size vascular bundle. So therefore option C is the right answer here. So this was about this session wherein we learned about the dicot and the monocot leaf and some of the MCQs related to it, which is important from exam point of view. So this was about the session. So we shall meet again in the coming session, taking up a new chapter and discussing the MCQs under it. So see you in the next session. Thank you.